would like to give you an introduction about what a complement system is and the different pathways which is involved in the complement system. So basically the complement system, it's a part of the immune system, innate immune system, which aids in the lysis of pathogens using a series of complement proteins. Now out of the 20 plasma proteins, there are 11 proteins which are mostly involved in the complement system, which includes B, C1 to C9 and D proteins, which are mainly enzyme precursors which catalyze a series of enzymatic reactions to form the final products of the complement system, which will eventually have various immune effects. Now this complement system, it consists of three different pathways: the classical pathway, lectin pathway and the alternative pathway. Today I'll be discussing about the classical pathway, which is an antibody dependent pathway. Now suppose we have a pathogen or a microbe in circulation, and to launch a complement system, the antibody has to come and attach to the surface of the cell surface of the antigen, that is our microbial cell or the pathogen. And mostly IgG and IgM are the antibodies which are involved in this pathway. Now, as we have all studied while learning about the structure of an antibody, that the antibody has an Fc region or the Fc loop. Now, if we have an antibody that is floating freely, I mean to say if it is not bound to any antigen, then this Fc region or Fc loop will not be exposed, and thereby it cannot form or it cannot activate the complement system. But when this antibody is complexes with an antigen, that is when it is bound to an antigen, there will be a change in the Fc region conformation of the antibody, which will expose the Fc loop. And this Fc loop is the binding site for the C1 protein, which is the first complement protein in the cascade. Thus, this C1 protein, it will consist of three regions, as you can see, this C1q, C1r and C1s. Basically two subunits of C1r and two subunits of C1s. So before I begin with the pathway, I would like you all to make note that the complement proteins which are involved in this complement system are in the inactive state initially. That is, they are not active before fixing the complement. And to make them active, these proteins are cleaved into two fragments, that is the A fragment and the B fragment. And usually in most cases, A is the larger fragment, that is inactive, and B is the smaller fragment, which is active. Okay, so as I told you earlier, when the Fc loop is exposed, that is when the C1 protein is the first protein that comes into action and it will bind to the Fc region of the antibody. When it binds, it will get activated and it will start the cleavage of other complement proteins. So first it cleaves the C4 protein, that is the C4 protein, into C4a and C4b. And then it will cleave the C2 protein into C2a and C2b. Then the C4b protein and C2a together it will combine to form a complex known as C3 convertase, that is C4b2a. Together, these both together are combining to form the C3 convertase. Now C3 convertase, as the name suggests, it will cleave the C3 protein into C3a and C3b. This C3b will combine with the C3 convertase, that is C4b2a and C3b will together combine to form another complex that is known as C5 convertase. C5 convertase, that is also C4b2a3b. C4b2a3b. Together it is combining to form the C5 convertase. This C5 convertase, again, as the name suggests, it will cleave the C5 protein into C5a and C5b. C5b will then recruit other complement proteins, that is C6, C7, C8, and form another complex that is known as C5b678. This is the complex C5b678. This is the complex which will be formed, which will be recruited by the C5b, and it will form a complex. Now this complex C5b678, it has the capability to polymerize the C9 molecules. As you can see, these are the polymers of C9, which will form a pore in the cell membrane of the bacteria or the pathogen. And this complex is called as the membrane attack complex, or MAC. And through this pore, there will be an influx of calcium ions and many other electrolytes into the target cell which is the microbial cell here, which will eventually bring about bursting of cell through osmotic lysis. So this is how the complement system works with the classical pathway. And my references are, I referred the textbook of microbiology by Anant Narayan and Panikkar, 7th edition.